Okay, they're calling this brand new chemistry, brand new science, and I agree. Enzymes are the product of bacteria. All right, your body has thousands and thousands and thousands of different single-celled bacteria. And what does a single-cell bacteria do? Its purpose in life is to create a very, very exotic, complex enzyme. You see what enzymes are? Enzymes have a molecular mass of 20 to 100,000 atomic mass units. You know what an atomic mass unit is? It's a proton. And in my world, a proton is 1,835 electrons. So multiply 1,835 times these numbers, and that's how sophisticated these enzymes are. And they come out in these strands, like bazillion long, just like that, just like that. They squirt them all out, and then all of a sudden they go just like that. And then they're stuck in that exact, specific, exact chemical reaction. If you just went like this, it doesn't work anymore. It's denatured. It has to be precisely like that. And what happens, it clicks with a whole set of other molecules, and every one of them at the same time changes at the speed of light, because the click is like that. When I did that, every one of these changed instantaneously, a little bit of their polarity. And when it did that to this other molecules, they turned into whatever they were supposed to, instantaneously. Listen to this. Enzymes can increase reaction rates by an enormous factor, up to 10 to the 17th power. That's 10 with 17 zeros. Faster than it would happen with an uncatalyzed rate. It would happen, but it would just take forever. So they tend to be extremely, not just very, extremely specific. Good morning, my wonderful friends. Today I have some very, very interesting information about the human microbiome, which I've been studying for many years now because of chemistry. I'm a chemist. I do a lot of things, but chemistry is one of the main important things. And now that I understand the nature of particles that make up matter, I understand how catalysts work. Nobody's ever been able to figure this out. A catalyst is an enzyme. An enzyme is created by bacteria. Well, what that enzyme does is it creates an instantaneous change of thousands and thousands and thousands of molecules instantly. Boom! And I know why now. And it's because of what is now termed click chemistry, where magnetically an invading molecule touches a whole series of particles, and instantaneously at the speed of light they all change. Okay, this makes it totally simple and easy to understand. What's the difference between bacteria, enzymes, and catalysts? Well, unlike bacteria, which are living things, enzymes are not living things. They're little packages of chemistry, really. That's what it is. It's just like this thing, just like that. It's not alive, but it's got a potential to bond to something, causing it to change its molecular, you know, what it is as far as a molecule goes. So let's start again. Unlike bacteria, enzymes are not alive. They're little packages of magnetic particles. That's what they are, really. They neither grow nor reproduce. They stay exactly the way they are, or they don't function. Enzymes are typically generated. They're made by bacteria. Every bacteria creates an enzyme. That's where we got to be looking at, is the bacterias. What bacterias are in people that are sick and healthy. These bacteria serve as catalysts for anaerobic digestion, which means inside your body you're going out. Well, here's what it says here. Enzymes can be thought of as knives that chop down complex molecules into smaller pieces, which are more palatable for the bacteria. Well, that's nice. That's good. It breaks up stuff so that we can eat it and digest it and all that. But there's a lot more to it than that. All right, my friends, I'm just going to let this play for a couple of minutes. I got this from a friend, Andy, who is a bacteriologist. He runs 19 labs that test the food coming into our country to prove that it's edible. You know, it's not going to be making people sick. So he's very, very aware of this. He sends me a lot of things. Now, just listen to what they have to say. This is basically a new science. It's a new frontier in health. Listen.
I'm thrilled to be with you tonight to share with you a relatively new and I think incredibly exciting field of research that's really changing our view of human biology, the field of human microbiome research. And that really describes studying the intense, diverse communities of bacteria, fungi, and viruses that live in and on the human body and understanding how they shape our health. So what will we cover tonight? Well, first things first, how do you study entire communities of microbes? What tools do we have, and how do we understand not just who's there, but what they're doing and how...
All right, let me just stop it right there and interject what I want to propose as far as research goes. I'm not telling anybody to do anything whatsoever. I am not a doctor. I am not giving health, anything to do with health. I am a researcher. All right, I was given 2 strikes by YouTube for talking about probiotics during the pandemic era because they didn't want anybody talking about anything except that. So I was given 2 strikes, and if I get 3, my channel goes. So I'm really worried about speaking in this realm. I'm not telling anybody to do anything. I never did. I never did. But there was such a lockdown on information that you get a strike, boom, 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 just like that. Now, anyway, please, this is just research, and this is going to make a big difference. Because at this point, she says, how do you study entire populations of diverse microbes? Easily. I could show you how. We're going to study it right now. Okay, my friends, if you are scientific at all, and you understand at all about catalysts, it's just beyond belief, really, how quick these changes happen. They happen literally at the speed of light. Now, a catalyst is a substance that participates in a reaction. It participates in it. It's like a spark. It's like a jab. It causes it to occur more rapidly, and I mean rapidly, but it can be recovered unchanged. So the catalyst comes in, goes bang, and then it just comes back out, and everybody that it affected happened. And I'm talking about literally hundreds of thousands of particles instantaneously. And then that catalyst comes right away. It says it's recovered unchanged at the end of the reaction, and it's reused again to do somewhere else. Catalysts may also control which products are formed in a reaction. That's exactly what they do. They end up having a product that is a result of what's called click chemistry. And I'm going to explain to you what click chemistry is. This is also unknown because they don't understand dipole nature of the particles. They're all dipoles. And when you come in with the correct chemical signature, well, magnetic signature, and you go bang, everybody out there gets affected at the speed of light. All right, look at this. Nobody could possibly believe this. An enzyme increases reaction rates enormous factors, up to 10 to the 17th times. That's 10 with 17 zeros. Faster than if you didn't catalyze it. It would do the same thing. Eventually it would turn into that stuff, but it would take a bazillion years compared to, I mean, literally within an instant, a second. And they tend to be very specific. That's right, because they're making an exact product. Now, what is it that the bacteria do? The bacteria squirt out little magnetic particles, virtually just like this, very fast. All right, and then they go like that, only they're very, very complicated, extremely complicated. They have, oh, thousands and thousands and thousands of these particles, not just like this. I think it's 20,000 or so. And then it's exactly like this, never going to be any different than that if it works. If it went like this, it wouldn't work anymore. It'd have to be exactly the way it was when it was produced and locked in as a protein. Then every time it hits any of these other magnetic particles, because everything's magnetic, it hits it and instantaneously it changes up to 10 to the 17th power of all of those done at the speed of light. That's what click chemistry is, and that's what magnetic chemistry is, and that's all there is now is magnetic chemistry. And if you understand it, you understand the energy releases and the heats and the reaction times and the products, and you know how life really creates these enzymes. And these are the only thing, these enzymes right here, that's the only thing in your body, the only thing that creates products and protects you. So what do we have to do? We have to figure out what bacteria is missing, because every one of them makes an enzyme. They are the factories for enzymes. We need to do a complete bacterial analysis on the human body of like zillions of people, and then just lay it out in like spreadsheet stuff, and you'll see everybody has diabetes. You know, not everybody, but a lot of them are going to fall into this pattern of these bacteria are not right. Something's wrong. And people, you know, because they talk about even like fecal transplant, it does not work for Crohn's disease. That's what they say. Now, Crohn's disease, I think I understand why it wouldn't work for that, but that's a whole other issue. When they do a fecal transplant, it puts new bacteria into you, and they start creating new enzymes. And enzymes are working. Not 92%. Well, hold on one second. Okay, here she is talking about it. Now, this is not my work. I'm not saying this, or I'm not making any recommendations. They're talking about a study they did with kids and fecal transplants. Now, listen.
There they saw significant reductions in neurobehavioral symptomology in those children. In this case, instead of a single treatment where there's a colonoscopic delivery of the fecal slurry into the diseased gut. Wait
a minute, before we go any further, just let me come back here. Let me start a little earlier on this, because this is about, they call it yellow soup. What it is somebody else's poop put into your body, somebody that's extremely healthy. Now, I'm not recommending this. This is not me. They're doing this research at UC Berkeley or wherever it was. It's not me whatsoever, so don't come after me. All right, they recently rediscovered fecal microbiome transplants. All right, so here they go. They recently rediscovered them. So here goes.
Discovered fecal microbial transplant. And what's very exciting is that it is essentially doing what we do in the mice, but instead it's transferring the healthy gut microbiome from a healthy donor to the gut microbiome of a patient with a disease or a condition or an infection to try and reconstitute the gut microbiome of the patient and treat the disease. So this has 92% efficacy in patients with Clostridium difficile infection. Antimicrobial treatment for Clostridium difficile is about 30% effective. And in this trial that used fecal microbial transplant to treat Clostridium difficile infection, they actually stopped the trial early because it was really not, they couldn't not treat the patients with this treatment because they were seeing 92% efficacy versus 30% in the vancomycin taper antimicrobial treated patients. It was unethical to continue the trial and not use this to treat patients. And we actually offer this at UCSF as a treatment for Clostridium difficile infection. It's also been used in a small pilot early study of children with autism spectrum disorder.
This is really interesting because I know of someone over in Ireland, Marguerite O'Reilly, who is heavily, I worked with her for years, and her son had terrible, terrible autism spectrum disorder. He was nonverbal and all kinds of things. Hopefully she'll comment on this, and I'll put her comment up as a pinned post. But again, this is not me. I don't do this. I don't know anybody that has autism. I haven't said take this, take that, whatever. But when I started working with her, she said all the kids that have autism have problems with their gut. And I said, well, and then we started talking. But anyway, let's just go after this here. So she's talking about fecal transplants and autism spectrum disorder. And this, again, not me.
It's about 18 or 19 children in the study. There they saw significant reductions in neurobehavioral symptomology in those children.
When they say significant reductions, that's important to know who was saying that. All right, these are researchers.
In this case, instead of a single treatment where there's a colonoscopic delivery of the fecal slurry into the diseased gut, they did that initially, but then they followed it up with a month of sustained microbial pressure in which the children actually consumed freeze-dried fecal capsules. And that was sufficient, a month of treatment, to significantly reduce the neurobehavioral deficiencies in these patients. They've also recently followed up 2 years later with these children, and this effect is sustained. And in fact, they've seen even greater improvements across this small cohort that has been treated.
Wow.
And so now this is under clinical trial in much larger placebo-controlled studies across the country as a potential treatment for autism spectrum disorder. We at UCSF have been looking at inflammatory bowel disease. I'm in the gastroenterology division. I can tell you that we looked at Crohn's disease and ulcerative colitis, and fecal microbial transplant does not work for Crohn's disease, at least the way that we tried it with a single colonoscopic delivery. We had several adverse events and we shut down the study. And
well, I'm not going to comment on this. All right, here goes.
I think it's important for that message to get out, equally as the 92% Clostridium difficile efficacy message. It's not equal. The microbiome is not the answer to all of our patients' ailments. And I think that it's important that we're cautious and that we are careful about how we implement this field and how we use this field to treat our patients.
I agree 100%. But I do want you to understand there is a new, well, let me just show you the results of my research on what I am going to be calling the immunothelium. And it is a layer just below the membrane that houses the enzymes. And the enzymes are the product of the bacteria. And the bacteria live in us in copious, diverse quantities. And well, that's what I would like to see, is studies on how much bacteria, where it specifically resides, because it may not be everywhere. Because you do have it in your colon, is absolutely 100% through your digestive system. It's just saturated with this stuff. And that's how it floats around, I believe. But there's a lot of study to do here. She's right. She's 100% correct what she's talking about. All right? Susan Lynch, PhD, UCSF, Division of Gastrointestinology. And now, I believe I have stuff to add, so here comes what I have to add. And again, I'm not saying do this, do that. I'm a researcher. Okay, so we know about the bacteria versus the enzymes and their catalysts and how they work to break down food versus also to attack invaders. Now, I'm going to show you where this immunothelium is and how it works. Okay, I do research into fossilized remains, and I have seen this layer in the fossil remains that I found, and these little tiny balls, which are called an interstitium. Now, this is basically skin, or the outside layer of your body, or an organ, and then there's a membrane, and then there is this layer I'm going to call the immunothelium. And then you get down into the interstitium, which allows you to move around. Now let's take a look at this in deep detail. Oh, by the way, this just came out in 2018. They just are calling this a newfound organ, and I agree with that, no question whatsoever. This is your immunothelium. Now they're talking about the interstitium, which they never had seen before either. By the time they saw them in slides, they were flat as a pancake, and they were all prepped and everything. And they said, see, previously researchers had thought these tissue layers were a dense wall of collagen, strong structural protein, which they are, but the new finding reveals rather than a wall, the tissue is more like an open fluid-filled highway. They always had them flat and worked with and crushed and put chemicals in them and then sliced them thin as they could be. See, look. Researchers said that filled spaces had been missed for decades because they don't show up in the standard microscopic slides where they crush them and do all the chemicals and all that business. All right, here's the deal with the interstitium. This is the layer of membrane. Now, it could be your skin. It could be the coating of a lung. Everything has a membrane that separates it from the other tissue that it's not supposed to be growing into. That's the key. A membrane is a separation layer. Now, how does that work? We know you have to absorb things in here, so it has to go both ways. When that food is going down through your intestine, you got to take some of that stuff out of that food and bring it in. Well, how do you do that? There's a layer right here, and this is what I'm going to call the immunothelium. That layer is a fluid-filled highway, right there. Then below that, you come into these fluid-filled spaces and these springs. They're really springs, and these balls. The balls attach into your body, and the floppy stuff lets you move around, but you come back. These are like springy springs. So you can pinch them, and that thing will go flat like this, and then bloop, it'll come right back to where it's supposed to be, because the ball will bring it back, the spring will open back up. And that's because these are different types of collagen. One's a very floppy collagen, the other one is a springy collagen. This will be a different type of collagen. Your skin is a different type of collagen. I think there's 28 collagens, and they all have a different chemistry. Like I said, floppy, stiff, springy, stretchy, not stretchy, all of that stuff. And that's why tendons are not stretchy to speak of. They're very, very tight and tense, and then they spring, and there's all kinds of chemistry going on in your body related to the tissue types. Let's go with that. And collagen basically makes up almost all the tissues in your body, except your fleshy stuff, the bloody, fluidy stuff, and that all runs out, and you end up with these. That's why they were looking at these mats of dense collagen. That's what they were looking at, collagen bundles. Now, that is the new organ that they found. They didn't even know this stuff existed before. I was looking at this for years, thinking, what the, and I talked to a guy about it, and it was really very, very accomplished anatomist. I won't use his name. I'm going to stop using people's names. I may use their first name, but some first names are so specific that they're still going to get you. So he's an anatomist, he's PhD, he's very, very accomplished. And he was the only one I could find that knew anything about fascia. And when I first talked to him, he told me that they used to call it fuzz. They just thought it was fuzz, because it would come out webby, gooey, like fuzzy. And he said they didn't think it had any meaning. And they would take it out and cut it out and sort of just discard it. And that is what this is. And it's more meaningful than you can possibly imagine. This right here does everything in your body as far as chemistry goes. And it is your immune system almost. That's what I'm saying it looks like to me. And again, I'm not telling you to do anything about it. I'm just saying that's what my research looks like. Okay, now, this is the immunothelium that I'm talking about right here. Here it is. This is the same pictures. This is not quite as close as this one, and then it goes right down into the tissues. But we're looking at this spot right here. Blown up is this spot. What is this? What's going on there? You have no idea how many layers of skin and tissue and layers in between everything. This is not some rendition of this. This is the actual microscopic structure of a membrane. That's how much there is going on in that membrane. Now this layer right here, I believe this white layer between the membrane and all of the tissuey stuff, because this starts to get into tissuey, and then it gets more and more into the flesh. But right here is that membrane of those balls and straps, and they're going to be shown down here in a second. But at any rate, this white is the fluid-filled highway, and that's where those enzymes go. And I can actually show you those. It's amazing what's come out just recently. Now, this is, again, that's the balls. Down here is where the balls are attached to the straps, and they're all tangled together. And this is what the balls look like themselves. Now, that's biology. Now, you may not be able to see this well, but there's also these membranes are very well defined. And there is some kind of a different collagen than stuff down here. Everything's different. It's separated. One side's separated from whatever that is, from whatever this is. And this is how specific this, and how fine and detailed this stuff is. You really got to do some work here. Well, you don't have to do a lot of work. You just got to be interested and start digging around. It's very simple to do this kind of research now. I mean, I have, see this here? I bought all the stuff. I have all the, and that thing is powerful, that microscope, and it's a waste of money. Because I could do this right here. I didn't do that. Somebody else did that. And it's all over the internet now, so you don't have to do that. This is what you want. You want one of these. Now, I had to make this thing real long myself. Normally, I'd only go about this far. I don't know why. But now, all you do is take a copper tube the same size as this. And this is the one that I love this thing. That sucker there does fabulous work. Anyway, because you could see way off from the distance and come down right onto it close. You can't see this fine. Well, I don't need that. I can go on the internet and see that. I'm going to teach you about catalysts and enzymes and bacteria and so forth. This is what happens when they squirt out, these bacteria squirt out these little particles. Well, what is that? And then they turn into these. Well, here's how they do it. At a certain point, they start to just snap together. This is called click chemistry. They click into a pattern of all kinds of really I mean, that's pretty exotic. And if they're not exactly like that, they become denatured. That means its natural state is like this. That means I'm going to click on here, I'm going to click here, click here, click here, click, 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 click. There's probably 1,000 different clicks that have to match up in order to make a certain substance. If you just go like this, that's it, it's done. It's not going to happen. That's called denatured. But this is these just squirt right out of the bacteria. So if you need something done, and one of these enzymes can create something at 10 to the 17th power per second, you better have good bacteria to make this stuff, because if the bacteria is not there, you're not going to have the enzyme. You don't have the enzyme, you're not going to have the chemistry. You don't have the chemistry, you're not going to break down the food. You're not going to be able to attack the invading bacteria. And there is, I show, it's called, well, here, I'll show the damn thing real quick. Okay, this is from Seeker. Very good channel. This guy is Gokul, and he's at one of the big universities in California. And he's saying right here, we are in a new era of cell biology. For centuries, microscopes have illuminated previously invisible worlds, and the recent advancements in microscopes are stunning. Where do you see, whoops, where do you see what he has to say here? The title of this is Your Textbooks Are Wrong. This is what cells actually look like. Now, listen to what he has to say. He's a nice guy, and he's looking for answers. Here it goes. Listen.
Within each cell.
This is an unprecedented view of the cellular world, where we can actually see immune cells scooping up sugars in the ear of a zebrafish in real time.
Focusing only on the crawling immune cells, we've noticed two classes of them. One was not hungry at all, but it was very active in terms of trying to figure out, what the environment is. There's another one that was kind of slouching around with a lot of food in its belly. We can actually conceptualize and visualize and analyze the contents of each of these cellular compartments in this crawling immune cell as it's kind of, scooping up its environment. I mean, that is a level of details no one's seen before.
We're living in a new era of cell biology.
All right, you should come up and watch this. It's called Your Textbooks Are Wrong. This is what cells actually look like. It's only like 8 minutes long, but that's enough to show you. These things are not understood. Let me just put it that way. And right now, I do feel like I understand from the research I've done how these enzymes are created from these bacteria. And I'm sure that different bacteria live in different areas. The lymph nodes are specifically something that I want to focus in on because they are the dumps of your body. That's where all the trash is brought to. That's where all the toxins end up. If you can't get rid of those, you're in trouble. That's where you get the lymphatic cancer. And that's very, very serious. And I think it's strictly a thing that you can't break it down. That's what these enzymes are for, to break things down. I'd like to have the lymph nodes tested for different bacteria. That would be the place I'd first start. And you could do them from autopsies, you can do them from, they remove them. They take them out because they're swelling up and they're going bad. And they think if they just take them out, everything will be fine. Well, maybe. But I would like to see what bacteria is living in there. All right, again, this is not me. The human microbiome, a new frontier in health, a watershed moment in human biology. And this is all from gut research they're talking about. And they want to know, how should we proceed? Well, here's what I would recommend. Find out exactly what bacteria is in everybody's body. Check it from the poop. Check it from the interstitial layer there, the immunothelium, where I say all these enzymes are residing. Get a complete set. Find out specific regions, kidneys, lungs, all that stuff, because they're going to have different bacteria, because they're going to have different products that are needed to function and to like in your intestines, in your bowels and so forth. You're going to have all kinds of different bacteria there to break down different foods. You're not just going to have one bacteria that goes up and breaks down everything. No. You're going to have all kinds of stuff. And I believe now, I honestly believe this, I'm not kidding you, that your tonsils might be the things that tell your bacteria down in your gut to get ready. You just ate a pizza or something. I'm not kidding you. Otherwise, you just sort of got to deal with it when it gets there. Maybe, I don't know. What does the tonsils do? They say they're nothing, they take them out. Anyway, this is what I mean. These are huge rabbit holes. Now, what would we need after we had that bacteria set of everybody? Say, well, you got all kinds of this lactobacillus, da-da-da-da, and all kinds. Now, but you don't have this, and you don't have this, and you don't have this. You find out what kind of diseases. So you put in the bacteria that everybody has, and the diseases that everybody has, and you're going to end up probably finding that some of them at least, there'll be bacteria that just is dead. It's not there, or it's very, very weak, and you end up with some form of disease because your body can't get the necessary nutrition or can't protect itself. Now, how would we find out what bacteria there are? I think you could get them out of autopsy samples. They're going to be resident in there. I don't know how long they'll last, but you could probably get them out of there. And you can get them from poop. I mean, there's no, you're not doing any invasion from poop. You're just getting a poop sample and saying what's in that. And you'll probably see daily it'll change. Because it depends on what you eat, really, as far as I'm concerned. You may have to do it over and over and over a bunch of times. And maybe you could even try doing it with different foods. You know, there's different foods versus the bacteria. Because right now I'm a little concerned about some of the pesticides they use, because they've engineered the food not to be damaged by the pesticide. But once you eat that pesticide and it gets inside of you, does it damage the bacteria that's in you? Because that's your weeds. that's your, you know, growing life that's in you that has to be not damaged by these pesticides. Anyway, or herbicides, I'm sorry. Because this is nothing more than, bacteria is nothing more than like plant life. Basically, that's what it is. And so if you got something that damages plant life and you're eating it, well, that's what I'd like to see. Whole, you know, natural foods versus, you know, farm processed foods, you know, the big food processors, because they use some techniques that could, you know, who knows. We want to know this. I'm not trying to hurt anybody or do any damage to anybody whatsoever. You know, we have to look into these things. Now, then we would have to check supplements that you take versus the results you get. And then all of this stuff laid out in the database. And I'm hoping that I'll be able to discuss with them, because I have as much research on this as anybody, to be honest with you. And Marguerite has even more research on this, because she was intimately, and she's got a whole program going now over in Belfast in Ireland, because they saw the same results that she's talking about. So, I love you all. Watershed moment in human history and in biology and based about everything.